We first present how insulin's receptor works and then the action of insulin on muscle, liver, and fat cells. If you haven't seen how insulin is released from the pancreas, go ahead and see it here. Insulin that was released by beta cells travels from pancreatic veins into the portal blood, making its way to the liver, where over half is removed from the bloodstream. The remaining insulin continues traversing through the systemic circulation, eventually reaching target cells. Insulin binds to the extracellular domain of the insulin receptor, a tyrosine kinase receptor positioned on the plasma membrane. The receptor is composed of two extracellular alpha subunits and two beta subunits whose parts are found extracellularly, traversing the plasma membrane, and extending intracellularly. When insulin binds to the extracellular alpha subunits, it triggers autophosphorylation of the beta subunits. This activation enables the intracellular tyrosine kinase domain of the beta chains to phosphorylate tyrosine residues on enzymes, including the insulin receptor substrates. The insulin receptor substrate functions as a docking protein, allowing the binding of several other downstream proteins to it, which in turn activate a number of pathways and enzymes depending on which tissue it is, something that we're about to see. Let's start off with how insulin affects skeletal muscle. When insulin binds to the insulin receptor, it activates it, and in turn downstream proteins are activated, just like we talked about earlier. This downstream signal mobilizes GLUT4 containing vesicles, which now fuse with the cell membrane, and the GLUT4 transporters get inserted in the cell membrane. Now, glucose molecules can enter the cell. Because of this, the glucose concentration in the blood decreases. That is, your blood sugar goes down. Importantly though, note that these transporters are GLUT4, not the GLUT2 transporters that are found in liver hepatocytes and pancreatic beta cells. The glucose that has entered can be converted to glycogen, and actually, insulin ups the activity of glycogen synthase, promoting glycogen formation. Moreover, insulin also stimulates enzymes involved in glycolysis and eventually the generation of ATP. In summary, insulin has opened the gate for glucose, which can then be used up or stored. Insulin also promotes protein deposition in muscle by increasing the uptake of certain amino acids, increasing transcription, and increasing the translation of mRNA. We proceed to see how insulin affects the liver now. Glucose molecules can enter the liver hepatocyte via the GLUT2 transporter. Remember, not the one that responds to insulin. Just like in muscle, however, insulin activates enzymes involved in glycogen synthesis, including glycogen synthase. And so insulin lowers blood glucose concentration by storing it as glycogen in the liver. Insulin also inhibits gluconeogenesis, that is, the formation of glucose molecules, and stimulates enzymes involved in glycolysis, eventually yielding acetyl-CoA. Some of the acetyl-CoA, along with oxaloacetate, forms citrate by the action of the enzyme citrate synthase. That's the first bit of the citric acid cycle for those who have done their biochem. Anyway, the citrate leaves the mitochondrion and is broken down back into acetyl-CoA and oxaloacetate by citrate lyase. The acetyl-CoA is then converted to malonyl-CoA. That malonyl-CoA can go up to the mitochondrion and inhibit the carnitine shuttle, which normally allows the transport of fatty acids into the mitochondrium for them to be broken down. But now, because it is inhibited, less fatty acids can be broken down. Moreover, malonyl-CoA, as well as acetyl-CoA, go on to form more fatty acids, which associate with glycerol 3-phosphate to form triacylglycerides. The triacylglycerides can be packaged into lipoproteins that is nascent VLDL, check out our lipoprotein video, for transport into other tissues. Insulin, just like in muscle, also promotes the deposition of protein within the liver hepatocytes. Lastly, let's find out what insulin does to fat cells, that is, adipocytes. Just like in muscle, insulin allows glucose transporters to be inserted in the cell membrane, allowing glucose to exit the bloodstream and enter the adipose cell. The majority of the glucose undergoes glycolysis, forming two intermediates of glycolysis, dihydroxyacetone phosphate as well as glyceraldehyde 3-phosphate. The dihydroxyacetone phosphate is converted to glycerol 3-phosphate. Meanwhile, glyceraldehyde 3-phosphate undergoes subsequent reactions of glycolysis, eventually yielding acetyl-CoA. Just like we saw in the liver, these guys end up forming fatty acids. The glycerol 3-phosphate and the fatty acids associate with each other, forming triglycerides, which are stored in the adipocytes' lipid droplets. Insulin also affects the activity of two super important enzymes to know: hormone-sensitive lipase and lipoprotein lipase. Because it is a lipase, hormone-sensitive lipase, when active, under conditions of hunger, breaks down triglycerides into fatty acids and glycerol, which can then leave the adipose cell. However, insulin decreases the activity of hormone-sensitive lipase. Now, triglycerides can't be broken down in fat cells, and they remain in and accumulate in the adipocyte. Moreover, insulin increases the activity of lipoprotein lipase, which normally breaks down triglycerides from lipoproteins into fatty acids and monoacyl glycerol, that is, a fatty acid linked to a glycerol. Please see our excellent top-notch video on lipoproteins for more info. These components are then put back together in the adipocyte to form the triglyceride again, which is stored. Thus, overall, insulin helps glucose get into the adipose cell and increases deposition of fat in the fat cells. We would include a summary of the actions of insulin here, but you can just rewatch the whole thing sped up. If you have any questions whatsoever, please ask away. We all here at All Night Med would genuinely love to help you all.